Speaker for the session is Dr. Nikhil Gandasi, who will talk on maintenance of glucose homeostasis in hyper and hypoglycemic states in the cellular context. Over to you.
Thank you very much for giving me this opportunity. Thanks to Sudipto for inviting me. What better time than after Dr. Shashikala's talk, because it tries to put things into context and going into cell biology, that's what we want. I'm not going to introduce regarding endocrine pancreas. I think this audience knows that very well. So what I'm going to introduce further is that in the pancreas, in the beta cells, there's glucose that goes in, and in this process, once the glucose uptake happens, there's an increase in the calcium, and that is what leads to insulin secretion. And insulin is stored in large dense core vesicles. So these vesicles package insulin as the core of them, which you see in the cytosol here in the pink background. And then these have to fuse to the plasma membrane to release its content. And this is what happens in neuronal cells when the synaptic transmission happens. This is exactly same in case of beta cells as well. Peter Thorne and colleagues have shown this also in the human islets. I'll just go back and try to say what we are doing. What we are trying to do is to see this in a microscopy setting, trying to see these individual cells and what they do, and how we can visualize these vesicles, and how we can visualize the signaling behind glucose-stimulated insulin secretion. So one of them is a turf micrograph showing there with insulin granules on the left that you see there. So these are fluorescently labeled, and we can study individual molecules of different proteins. In this case it's syntaxin, in case of turf. So we are just seeing at the plasma membrane all these vesicles and also the proteins that are present there. And then comparatively with a confocal microscope, which most people do, you can also see these insulin granules in the periphery, and this is where they are generated in the cytoplasm and they come to the membrane. And this is what we are interested in, to study trafficking in terms of insulin secretion. So our lab focuses not only on this, it focuses on a lot of other things as well. We study G protein coupled receptors and drug targets and things like that. We study exosomes, which I'm going to touch upon a little bit. We study pancreatic aging, we study pancreatitis as well, and we study exocrine pancreas. So some of these parts with exosome secretion, exocrine pancreas, pancreatitis, and also glucose-stimulated insulin secretion, will have talks tomorrow from my students who are all there in the audience. So to start with, when you look at a turf micrograph and when you try to look at these and then stimulate with glucose, what happens is this. I hope I can play this. Well, this doesn't play. So what happens here?
Just put the escape button here. No, just escape that. Escape this. Just press escape. Yeah. Now you can just move on to that. Yeah, it will be great.
It's fine. In the interest of time, I'll just go forward. So what happens there is that the insulin granules which are present there, they fuse something like this. You can see the individual events there at the bottom. You see that they brighten up, because this is basically a fluorophore thing. Because they are acidic, then they undergo an increase in fluorescence, and then they disappear, which is what is exocytosis. What is interesting for us is that whatever is exocytosed, there should always be individual granules coming from the cytoplasm of the cell to replenish this. So this is a constant supply that needs to be maintained, and this is supplied by something called docking, where the individual granules which are present in the cytoplasm are coming to the plasma membrane of the cell there. So to study this is what we were interested in. So we had access to human islets when I was back in Gothenburg, later in Uppsala, and also in Oxford. So we collected these human islets and we tried to stimulate these cells individually, like I showed in the previous slide. So we tried to see the total number of dock granules and how many of them do undergo exocytosis. So when you try to correlate them, you try to see the number of dots here, which is the dock granules, and then you try to see how many of them undergo exocytosis, which is on the x-axis here. So these are individual donors that you see. In these individual donors, what you see is that there is a very nice correlation between the number of dock granules versus the total number of exocytotic events that you see there. Interestingly, there is a small sort of data points that you see there at the bottom, which is the type 2 diabetic ones, and where we see lesser number of dock granules. And this is what we try to depict. Again, individual donors here, and you see consistently that total number of dock granules in case of type 2 diabetes is decreased a lot. So in this sense, what does this mean? this all means that we study a lot of, we try to measure blood glucose, we try to see that in the peripheral blood, and this is what we see, which is a biphasic response. And in type 2s, we don't see that. So we wanted to see this at the single cell level, at the single beta cell level. So using microscopy, we did exactly that, and in this case, we can see that there is a biphasic response, like you see in the peripheral blood. But also, what is interesting is that the docked granules that are present there, they also lose out, which means that they are the ones which are important for the insulin secretion to proceed. What this all means is that there is something that is trying to push these docked granules. That is, in fact, glucose transporters themselves. We try to study this, Pallavi, who is among the audience, we try to study glucose transporters there, and in that case what we saw was that the total number of transporters recruited when these individual beta cells from humans are challenged by glucose is that the transporters don't get recruited to the membrane, which means that there's also one of the reasons why the insulin secretion is decreased. So we also tried to study the expression. Expression is also decreased, but also the functionality is decreased. So this is a paper which got published a month ago in PNAS, so I encourage you to look at that. So in that, what we tried to describe is that there are two problems. One is that the transporters being recruited to the membrane, which is decreased in case of type 2 diabetes. We have mechanisms there. I'm sure Pallavi will speak about it, so I'll not take the show away from her. And also on top of it, there's also insulin secretion, which is decreased, which I had already alluded on to before. So what all this means? Can we use this as a treatment strategy? That is the question we wanted to ask. One of the postdocs in the lab, Saptadeepa, so she tried to see with several small molecules. One of them is a phytochemical here, furobidase. When she tried to treat these cells with furobidase, the transporters, which normally undergo a constant recycling, they're just captured at the membrane, which means that there is more glucose uptake that takes place because of this, which also means that we can increase the insulin secretion at the individual cell level, at the individual beta cell level. We have to test this at various settings and animal models and later humans and things like that, but that's a start is what we think there. So with that, what I'm trying to say with this part is that there's glucose transporters which helps in recruitment to the membrane, which means that these can get in glucose and that can lead to secretion of insulin, provided that there are enough insulin granules present at the plasma membrane of the cells, which can lead to biphasic response. And there are transporter related small molecules which can try to make sure that there are more transporters available, let's say in a hypoglycemic state. So all this is also that these large dense core vesicles, there's a lot of molecules involved. There is a sort of individual nanodomain that develops there with a lot of molecules. And these molecules were what we were interested in to see what these molecules do and how are they compromised in case of type 2 diabetes. Some of them I'll show you. Some of them is RIM1, RAB and all these. RAB is a protein which is on the vesicles that can bind to RIM which is present at the plasma membrane. So we do see that in case of type 2 diabetic individuals, so again human data, we see that there is less of association of the insulin granules with these proteins present there. Also the expression is decreased, which I will then later show you. But then before going into that, the formation of a nanodomain, which is important at the plasma membrane, which brings in vesicles, that being compromised means that there is less insulin secretion that takes place is what I'm trying to say. So we try to take these beta cells from type 2 diabetic donors, we try to overexpress some of these proteins, or some of them that I showed you in the previous slide. When we do that, what happens is that we can increase the number of docked granules in this case. So this we try to do and correlate this with also the secretion index of these individuals and also the expression of these proteins. You can see the hits are similar in case of B, and also in case of A you see that HbA1c inversely correlates with this thing where the expression is in case decreased when HbA1c is very high. So this is basically summarizing that. In the interest of time, I'll just go and say that there is glucose transporters that are involved and the insulin granules have to dock and then get ready with all these proteins that are present, leading to insulin secretion, and we do see that various levels this is abrogated. So we wanted to see how we can rescue it. One of the things that we were thinking of was ER stress. So in case of ER stress, what happens is that there are a lot of things that goes on. One of them is proteins which are not completely processed, which just gets stuck in this case granules. So what we wanted to do is to see what are the other things that can break this down. Amyloid is something that you might have heard of a lot. So these amyloids are processed proteins of insulin which doesn't actually completely mature. So these, can it be broken down? is one of the things that we were seeing. So there are two things, right? One is the functionality of beta cells, the other is keeping them alive. So in this case, if there are so many non-functional proteins, then keeping them alive becomes a problem. So in this case, what we're trying to do is to get rid of these proteins. So Priyadarshini, one of the people in the group, has been working on this. Multivesicular bodies is something that can break down these proteins. This has been known since 2018. We were trying to see in the cellular context, in case of beta cells, how this can be done. Is it there and what it's doing? It was the question here. So Priyadarshini initially tried to label these cells, which she was successful. Initially it was very difficult to label because these are very difficult to isolate and label. So she tried doing that and she was successful. You can see from the EM micrographs, when you label them as well, they are fine, they're doing okay and things like that. So with this as well, similar to the large dense core vesicles that contain insulin, also the exosomes or multivesicular bodies, they are also decreased in case of type 2 diabetes. Meaning to say that there is more amyloid present and the mechanisms that can break down the amyloid is also compromised. Meaning to say that also these are the cells which can undergo apoptosis. The total number of beta cells can then be decreased. That's the thing. So I'll just leave it at that because she's going to present that anyway tomorrow. And overall, we want to study these individual vesicles at the single beta cell level to see the answers and try to understand what is happening there. So apart from this, we also study other hormones. I said hypoglycemia, which is all linked to insulin. The other is hypoglycemia, which is more related to glucagon. We try to develop a marker there. And in glucagon as well, when we see the human donors, we do see a compromised glucagon secretion, which we see also a very nice correlation in this case with HbA1c. Again, the total number of glucagon granules in an alpha cell is massively decreased in case of diabetics. This is what is represented in the individuals that is plotted out here based on density and based on exocytosis and the HbA1c of those individuals. So meaning to say that there are a lot of possibilities in this. One is beta cells that we are trying to study, and there's alpha cells as well where there are massive possibilities but not explored much. I think there's a huge potential considering that people die out of hypoglycemia rather than hyperglycemia. So I think we should definitely try that. And also we try to do this all, trying to put this together in the form of islets as a whole to see what happens in the islets rather than single cells going back to the islets. And there as well we do see that there is compromises done on delta cells, which I don't have time to speak about right now, but just I'm trying to allude on to that. So overall, what this all means is that there is lack of human islets available in India, and that's what I want to bring to this platform, saying that creating a human islet network in this case would help us study lifestyle, genetic background and environment in the Indian context, which is something that we spoke about with Sudipto in the afternoon. I'm sure there is more possibilities with Dr. Shashikala and co on board. So I would like to thank all the funding agencies and the research group here, and I would encourage you to look forward to Pallavi, Meetu and Priyadarshini's talk tomorrow, who will speak more about all these things. Before I go off, these were the areas, and one final thing, basically an ad. We have a meeting which is happening in October 24th. There are posters at the registration desk. Please come over, and I'm sure this will be one more exciting meeting where we can meet and discuss. Thank you.
Thank you, Dr. Nikhil, for their excellent talk on the glucose homeostasis at the cellular context.
Any questions from the audience? We'll start with the audience.
This is a wonderful talk, and the depth of the islet biology work you are doing is excellent. But you said that you are working with islets coming from somewhere else, not Indian islets. And in the last thing you said that you want some Indian islets. We actually at AAG hospital, we do islet cell transplantation. We get islets from chronic pancreatitis patients. And we had earlier some cadaver donations and then multi-organ donor donations. But now I am also looking for, I have a grant, I said, no, Wellcome Trust has given a diabetic center under Clinical Public Health Research Center scheme. So we are also looking we are working with the government to get the pancreatic tissue for research. Because pancreatic tissue for research is not there in the law of Indian law, it's not there. So that's where we are also struggling. I think we are all should work together to get that permission quickly. Okay.
Absolutely, I think very well taken. I think those are the points which we have been thinking of for a while, and I think access to the tissue will make the understanding of pancreas in India very much more, so that we can try to see what are the differences that we see in our cohorts. Thank you.
Nikhil, fantastic talk. I was wondering, you showed that calcium is required for the exocytosis, right? So do we know the source of calcium, whether it is intracellular or extracellular?
So in this case, there is micromolar concentrations of calcium that is required. So it's extracellular calcium.
And
we have worked on that as well. It's L-type calcium channels is what we know right now, CAP 1.2. So we had started some things with 1.3, which my postdoc lab might have taken over.
Okay, thank you.
Yeah, the voltage sensitive channels, unlike the aspirin cell, that have crack channels that just put in a huge amount of calcium. Very good talk. I was wondering if you could tie together your thoughts after hearing the previous talks and hearing yourself speak. Where do you see the field going in the pancreatitis realm, and what kind of ideas do you have for us?
So with respect to pancreatitis, actually a very good question, thanks for that. So with pancreatitis, we have started some things which Meetu is going to speak about tomorrow. We also have these genetic landscapes that we have tried to see. I think what happens with these genetic landscapes is we also have novel ones which we see, but we don't understand the function of it in the sort of exocrine pancreas, which is what we are trying to do to a certain extent. And it becomes very difficult because somehow these exocrine pancreas, even when you isolate from the mouse, somehow you can't label them with something. It's not as easy as endocrine pancreas. I don't know why that is the case, because I would think the opposite, but that's not the case. But once we can get there, what we're trying to see is that one of the things that was told was regarding the channels, which also alluded on to. So this is one of the things we are interested in, because many of the variants that we see is related to the channels. So we want to see if these channels are recruited at the right place and how this matters for the lipase and amylase and things like that in terms of PDAC and things like that.
And how much of it do you think is due to the number of channels on the surface versus the regulation of the membrane potential?
So with respect to number of channels, when there is a depolarization and we reach minus 70 millivolt, we do see that there is around 20 channels that is recruited at the site or at the mouth of the channel, which is what you see as a cluster. But we also see that when you do patch clamp, there's 2 channels are good enough to still bring in micromolar concentrations of calcium, which is enough for exocytosis. So what 20 are doing, what 2 is, when 2 is enough, then I don't know that answer.
So the lack of calcium in some of the chronic pancreatitis islets, I'm wondering if it's an electrophysiologic problem, maybe having to do with potassium channels or some other leakage or hyperpolarization versus the channels themselves.
Very good question. We have actually been doing something in this regard, and we have published also something there. So what we see that expression of these channels are completely unaltered in case of type 2s at least. But what happens is the recruitment to the plasma membrane, which is dependent on a protein called MUNC13, which first recruits to the membrane and then brings over the channels, that has been compromised. With respect to MUNC13, there are multiple things that we see. It's a C2 domain protein, which is responsible for when there's an increase in calcium, that is when it goes to the membrane. And that is what brings in a lot of other things as well. And we do see compromises happening there. And we do think MUNC13 is the protein which can be one of the proteins which can try to bring in more and more channels there. So, but we don't have any targets in that terms. But happy to talk and show you the data what we have.
Okay, my last question is that when you have accumulation of dysfunctional insulin, are there deficiencies of some of the E3 ligases that are important for transporting misfolded proteins to the proteasomes, or have you looked at that?
We have not looked at that, but there are various research groups which have looked at it, and there is actually a dysfunction there. And these can also impact the snare proteins because there are transcription factors. So they also try to dysregulate the amount of proteins which are required later for the secretory machinery to